All right, so in this video, we're going to be talking about the ending of chapter 17, which is going to be on proteins as well as mutations. So just kind of want to remind you of the end process of this all. Once you make your polypeptide, it's going to undergo protein processing. So that's where you have your primary structure. That's where kind of this whole last bit was covered. So we talked about in the previous videos where you make your messenger RNA that goes out to the ribosome. Now we're going to be looking at just that last step where it folds. So there are things that help it in terms of how it folds. So this is showing you the first folding is the backbone with hydrogen bonds. It folds again according to the R groups. And then you have multiple polypeptides joined together. So that's going to be your quaternary structure. Now, I want to just kind of recap this chapter by showing you just kind of a summary of what the video was talking about. So remember to just to kind of play this whole thing out, you want to make sure you feel comfortable with what's going on in all facets of what's happening in protein synthesis. So just from the beginning, it's kind of good to see this whole video in one step. So here's my gene. This is RNA polymerase. So I'm actually doing transcription. This is my elongation phase. So I'm actually building my mRNA. And remind you guys that this is basically the elongation of my mRNA. So I'm reading the DNA to make my mRNA. If it's eukaryotic, we're still inside the nucleus, so this is going to undergo your RNA processing. So when you add these nucleotides, you're adding one at a time. But notice, inside the nucleus, the 5' gets a phosphate and a guanine. You're going to cut out the introns. Remember, introns stay in, exons exit. So those are SNRPs and spliceosomes. So this little loop of RNA is your intron. Now this is my coding segment called the exon. I add a bunch of adenines at the end, so the poly A tail. And then your mRNA is going to leave the nucleus. So that is transcription in the nucleus. Now, where does mRNA go? Well, remember, your messenger RNA has to go to a ribosome. Now, these things in green are the tRNAs. The thing in purple are the amino acids. So remember that your tRNA is going to be finding and searching the mRNA template until it hits that start codon, which is AUG. So there is your starting point. The larger subunit of the ribosome comes, and now we're going to be building your polypeptide. So remember, this is your protein that's being built. So the one amino acid bonds with the other, making a peptide bond. This empty tRNA is going to leave. And now we're going to basically be building this whole polypeptide here. So amino acid, I'm going to form a peptide bond right here. That gets shifted over. Another amino acid comes here. I'm going to form a peptide bond. Now this just kind of shows you the codon is going to be the RNA, right? So GAC is going to code with the anticodon of the tRNA. So this just shows you that an anticodon or a tRNA is going to pick up the amino acid, and that's how this whole process is going to basically get recycled. So remember, this is the protein. It's going to fold and do its job according to what the gene sequence was. So this would be your protein. Now this last part is showing you how this protein is processed, meaning how is it folded. So this is showing you something that I think is important. This ribosome is going to be on the rough ER. So I'm basically building and synthesizing a protein on the rough ER. Now remember, this is a throwback to the endomembrane system, right? It goes from the rough ER, travels on the cytoskeleton to the Golgi apparatus. This is where it gets additional help in folding. So this is, you're now seeing your secondary, tertiary, and quaternary folding. And then basically, if it is a quaternary structure or a protein that is shipped out of the cell, this is doing that exocytosis. So that last step is showing you how a protein is shipped outside of the cell. And so when you go back into this last step on proteins, let's just kind of remind you of the key things. Once it's folded, that is the functional protein. So just want to remind you, if a protein is made on a ribosome on the side as well, actually, let me, I should kind of write this out here too. So this question, describe the pathway a protein takes after it's made on a free ribosome to when it's transported out of the cell. So a couple of things. If it's a free ribosome, meaning that ribosome is not on the rough ER, the protein stays inside the cell. If it's going to be what's considered a bound ribosome, that protein is going to be shipped out to, I would either say the membrane or outside of the cell. So basically, this is just showing you if you have a free ribosome, it's going to go in terms of the endomembrane, the rough ER. If that protein is actually synthesized and actually goes to the rough ER, then it's going to be shipped out. But if it's a free ribosome, when we talk about free versus bound, we're talking about the ribosome. If the ribosome is actually on the rough ER, that protein is shipped out. If it's a free ribosome, that protein is going to stay inside the cell. So that kind of leads into what I need to wrap up on this chapter, which is going to be on mutations. Now, there are a couple of different types, but the key thing about mutations is do not think of these as being denatured. These are genetic issues. The mutation can be in the DNA or the RNA, but for the most part, it's going to be in the DNA if it's going to be heritable. But sometimes you do get mutations that happen when you're doing transcription. So when you think about mutations, let's just kind of jump back to this chart, your codon chart. So let's kind of remind you of what's happening here. So let's say our RNA was supposed to be GCU, right? So if the RNA was GCU, but let's say instead of, so let's say I had an RNA sequence GCU, and it was UUU, CCU, and let's say instead of this U right here, it was mutated to an A, right? So when I was doing my transcription, instead of being alanine, in terms of this, I had a mutation, GCU, I had a mutation, and instead of being GCU, it's now GCA. Notice it's still alanine. Well, that's what's going to be considered a silent mutation, but regardless of what we're getting into with mutations, if you change it, if you change the genetic sequence, you can, emphasis on can, you can change the amino acid sequence. So let's take a look here. What if I, instead of mutating it from a GCU, what if instead of the C in the middle position, what if that C is mutated to an A? Well, instead of being alanine, it would be this aspartic acid. So it just depends on the position. So let's kind of get into what mutations are. Mutations, the key thing is it can change the protein shape, right? And the biggest thing is that if the protein shape changes, it can drastically change the function. So we have what are considered point mutations. That's just where one base is going to be altered, so essentially one nucleotide. And then you have these larger ones where you have insertion or deletion, and both of those are going to fall underneath the category of a frame shift mutation. So we have a couple of different types we've got to address: silent, missense, and nonsense. So all of these are considered substitution. The first three we'll talk about, because we're just changing one nucleotide at a time. So let's kind of remind you of what's going to happen here. So we have what's considered our wild type, and remember, wild type just means normal. So if you look here, T would, if this is the DNA, if this is my template strand, T pairs with A pairs with U, C pairs with G. So this is my template strand. So we got transcription, translation. This is basically what it should look like. I should have methionine, lysine, and phenylalanine. Okay. Now notice right here we have an A, but if you look here, it was a G. So we have an A instead of a G. Okay. So now when you look at your codon sequence, it goes AUG. AAG still codes for lysine, UUU, and that's still the same. But now when we get to this last one, the original wild strand was GGC, coded for glycine. GGU also codes for glycine. We can get confirmation here. So we had that, whereas the GGU codes for glycine, GGC also codes for glycine. So when you think of it on this first example, the protein, the amino acid sequence is 100% the same. So it's 100% the same. So this is a nucleotide pair substitution, and we call it a silent mutation. No protein alteration because it codes for the same amino acid. Okay. Now let's look at the difference here. This one is going to be called a missense. So we have a T instead of an A. So we have a T instead of a C. I'm sorry. Okay. Right here, this gets mutated, and we now have a thymine in the DNA. Now, some of you might be asking, well, where did this mutation come from? It could have come from just when DNA was replicated. That's where most mutations stem from. So if I have a thymine instead of a cytosine, notice the first ones are still the same. I got AUG codes for methionine, AAG codes for this amino acid, UU, all good there. But now we get to this last one, we have AGC. AGC codes for serine, which is different than glycine. So the protein is different. Now, can this change the overall shape? It depends. If glycine is polar and serine is also polar, you're not going to have a huge change in your shape because they're going to have similar charges. But if glycine is polar and serine is non-polar and different charge, then yeah, your protein is probably going to have a different shape and different function. Just remember, if you change the shape, you're changing the function. So the reason why I mention that is some AP tests will say, well, glycine is substituted for another non-polar amino acid. If the charges and the polarities are the same, you're not going to have an overall big change in your overall protein, even though that is a mutation. So we call this a missense. Not a massive deal unless, like I mentioned earlier, if glycine was polar and serine was non-polar, then yeah, you would have a dramatic change in your shape. So these ones though, so those are basically the big ones. Silent, missense. The last one is going to be nonsense. So let's look at nonsense. Nonsense is actually a problem, a big problem, because notice right here we have an A instead of a T. So this was your wild type, we had a T. But notice now when you made your RNA sequencing, you had AUG and then you have a stop codon early. So that's what's considered a nonsense. So really it's termination of protein synthesis. So that's a nonsense mutation. Really you're stopping translation from happening. And again, all three of those fall underneath that category of non-substitution, where one nucleotide is changed, right? Now the last two, insertion and deletion, these ones have a much bigger problem because let's kind of revisit how your RNA is read. So remember that if this is your wild type, and if I actually had a nucleotide pair insertion or deletion, okay, whether you insert a new nucleotide or delete a new nucleotide, that's when we have what is called a frame shift. Now, notice here, if we have an A missing, so instead of being AAA, I actually lost this nucleotide. You might not think that's going to be a huge deal, but it actually is, because remember, your ribosomes read three nucleotides at a time, right? So if I drew in a ribosome, remember, it's reading in triplets. So instead of, it was AUG, AAG, UUU, GGC, and then you have the stop codon. The problem is when you deleted a nucleotide or added a nucleotide for that matter, in this case it's just showing you deletion, notice your next codon sequence is UUG, which codes for leucine. We have GCU, which codes for alanine. And you no longer are going to read a stop codon. That UAA is there, but notice that you're reading it in triplets, so your ribosome actually isn't going to be reading that UAA codon. So you're going to basically have a non-functioning massive protein because your stop codon isn't read. So whether you insert or delete it, that can cause some issues. Now look at here. This one is kind of interesting, where I actually removed a complete codon. So in terms of this, let's say if I took out TTC, if that part is actually gone, I took out 3 nucleotides. It still is considered a mutation, but it's not a frame shift. Why? Well, you've actually removed 3 nucleotides, so you're not actually having a frame shift mutation. And this just kind of shows you all kinds of, this is showing you the example of sickle cell anemia. So if you have that, it's just one single nucleotide sequence. This is that example that if glutamine was swapped out for valine, different structure of the protein. And the reason why is because glutamine and valine have 2 different chemical properties. So that had a big difference on the overall functioning of the protein. So kind of nice summary of transcription and translation. You're seeing all of this. This is kind of a nice summary. Remember, RNA polymerase is making your mRNA. You process it, right? So remember, exons are the coding segment. Exons exit, introns stay in. You add the poly A tail. That mRNA sequence goes out to the ribosome. You're feeding it in this direction, right? So you had, this is your growing protein, and that's going to be the end result. So make sure you feel you need to feel comfortable with the ins and outs of transcription translation, okay? Now, the last final thing that we need to mention is what's the difference between prokaryotes and eukaryotes in protein synthesis. And the biggest thing is that prokaryotes don't have a nucleus, and so transcription and translation happen at the same time. And the other big thing is that there's no mRNA processing. So that's a big one. They actually can't do any SNRPs and spliceosomes. They don't process it. So transcription and translation happen at the same time. So if you look here, eukaryotes on the right, they have the nucleus, so they make their mRNA, it leaves, you build your protein here. But on the prokaryotic cell, it still functions very, very similar in the sense that there's RNA polymerase. It still uses RNA polymerase. It still has a ribosome that reads in triplets. The genetic code is still the same. AUG starts the protein synthesis sequencing. It's just transcription and translation can actually happen at the same time. So just know that this is still the genetic sequencing for all cells, meaning if your mRNA sequence is put in, and this is what we get into in the next chapter, we can actually take a piece of DNA and put it into bacteria or a prokaryotic cell, and it'll express it because the code is universal, meaning AUG, regardless of the cell, codes for methionine. AAG codes for lysine, regardless of the cell type. So the genetic code is pretty interesting. So that is chapter 17 on protein synthesis, and we will see you guys in the next video.